Before all, let me put everything in the context. You know that the gut microbiome interact with the central nervous system through the gut microbiota brain axis. And recent studies show that imbalance in the gut microbiota may contribute to the pathogenesis of neurological disorders, including the Alzheimer disease. Alzheimer disease is clinically mainly uh, characterized by the accumulation of amyloid beta oligomers and tau protein deposition in several brain regions. Now, the concern is whether the buildup of amyloid beta and tau deposits are associated with the change in the human gut microbiota composition. And there's no well study actually. So, in this study, our goal is to assess the link between the gut microbiome composition and PET amyloid beta level, even PET amyloid PET tau level in the brain in Framingham Heart Study population. In fact, we use the participants cognitively healthy from the third generation, the neuroswim spouses, and the homicide cohort of the Framingham Heart Study who provides the stool sample and the PET imaging for neurological assessment during the third examination. And let me mention that the gut microbiome of each participant was quantified using the 16S sequencing method. After performing the mutual variable association and the differential analysis, adjusting for age, sex, and body mass, we found that the elevated measure of beta amyloid in the frontal, lateral, and retrospinal, and the elevated measure of tau, of PET tau, in the renal cortex and the inferior temporal are associated with the reduced abundance of general Butyricicoccus and Ruminococcus in the gut. And after performing the functional analysis, we found that Butyricicoccus and Ruminococcus are butyrate-producing bacteria harboring neuroprotective effect. This is globally what we did and we found in this study. About this question, we say the buildup of amyloid and tau in middle age, even in individual cognitively normal, are associated with the change in the gut microbiome structure and function. And another thing is say manipulate the abundance of bacteria harboring neuroprotective effect could help keeping the gut microbiome healthy. This can be the two main takeaways from the study. One very first step is maybe to understand the role that diet and medication intake play in this interaction. Then we should establish the causality and microbiome mechanism of the interaction using, for example, the longitudinal and the clinically international study. Another thing, I think we need to further explore the mechanism associated with the brain axis. We think that the gut-brain axis, we should also identify the different pathways through which the amyloid beta and tau could affect the gut homeostasis. I would say the main thing that clinician should keep in mind with this research, they should be aware that gut structure and function are intimately linked to brain health. Thus, clinical intervention that restore the gut homeostasis to result clinically in less amyloid beta and tau accumulation and for sure beta brain health.